Good morning, everyone. Today we'll be learning about neurons, neuroglia, and discuss about growth factors, nerve growth factors. In our body, we have different organ systems, near about 11 organ systems we have, like digestive system, skeletal system, muscular system, cardiovascular circulatory system, lymphatic system, integumentary system, skin, and digestive urinary system or renal system, endocrine system, reproductive system. So, nervous system. 11 different systems are there, and all these systems work through their organs. Now the activity function of these organs must be coordinated and they should be controlled. Now how their activities of all these systems are coordinated and controlled. So we have two different controlling mechanisms. One is by nervous system, neural mechanism, and another is endocrine mechanism by endocrine system. So these are done by, as I said, the nervous system and endocrine system. So nervous system, how does it acts? Nervous system acts by getting signals from the periphery, and it decides what has to be done. So accordingly, it does. It controls the activity of the other organs, means peripheral or its target organs. Okay, so there should be some efferent path that will be carry sensations from the periphery, and it must be having some efferent path through which it will be regulating the activities of all these organs. When it gets these informations from the periphery into this, various messages are formed in the brain, like our thoughts, memory, learning, feelings, movement and balance of the body. And with these informations, inputs, we can smell something, we can see something, we can hear something, we can taste something. So we can feel any touch, pressure, pain, hot, cold. So these type of sensations we feel, this feeling comes to our realization, that is through nervous system. Now let us see how these nervous systems are organized. So they have to control all these organ systems. How they are organized? So they are organized. The central nervous systems, as the nervous system, total nervous system is made up of central nervous system and peripheral nervous systems. Central nervous system is the brain. It consists of brain and the spinal cord. So these are the central nervous system. And the peripheral nervous system, some nerves come out of the brain and also from the spinal cord. These nerves are spinal nerves and cranial nerves. These nerves are spinal nerves and cranial nerves. See, this is the organization, central nervous system, brain and spinal cord, and this brain is sending or receiving some information from the periphery through these nerves, cranial nerves, and this is also part of the central nervous system, spinal cord. These are also having some afferent and efferent paths. These nerves are called spinal nerves, and these are called the nerves coming out of the cranial nerves. They form the peripheral nervous system. Okay, so now see the peripheral nervous system are of two types. One is somatic nervous system, another is autonomic nervous system. Somatic nervous system we can control voluntarily. Suppose I want to contract these muscles, So I want to breathe fast. So these are voluntary control. These are done by the somatic nervous system. So there are sensory divisions, and in the somatic nervous system there are sensory divisions and motor divisions. Okay. So suppose I have seen a snake. So this is the sensation I receive through my eyes. So now what I do? Generally what I do, I will run away. Okay, because it may bite me. So how I will run away? By contracting my muscles. So these are the voluntary actions. Okay, so this is somatic nervous system in brief, and later on we will be studying, where we will be studying in details all this nervous system, all this system, autonomic and somatic system. Then we will be knowing in details. Now, autonomic nervous system are of three types. In your books, you may find it as two types, sympathetic and parasympathetic. But we know there is another system, that is enteric nervous system. These are independent. Spontaneously, they can generate their own impulse, but they are again under the control of the central nervous system or autonomic nervous system. Now, let us see what is there in this nervous system, how this nervous system is composed of. This nervous system, what are the tissues, cells in the nervous system? So now in the nervous system we will get two types of cells. What are these? One is neuron, another glial cells, neuroglia. So two types of cells in the nervous system will get. What are these? Neurons and neuroglia or glial cells. Glia means adhesion. Glial, gum. So they will, what we do with gum? We fix something. So these glial cells basically fixes the neurons in its position. Moreover, that means it protects, it supports, and sometimes it also provides nutrients to the neurons. So they are of, we will see different kind of these glial cells or neuroglia. They are of different types. You will find there are 4, 5, 6 types of glial cells are there. So what every variety has specific functions. We will discuss about that. So nerve cells are of two types. One is neuron, another is, now everybody knows what is neuron. Structural and functional unit of nervous system. Two types of cells, neurons for processing. What are the functions of the neurons? They will process, transfer and store information. They will receive the information, they will transfer the information, they will receive the information, they will send the information necessary for necessary actions. So these are the functions of the neurons in short. And there are other cells, another group of cells, these are neuroglia. So they provide support, regulation and protection to the neurons. Okay, so these are the main functions of the neuroglia and main functions of the neuron. Now let us see what is in the structure of a neuron. So we know in the neuron what is there, mainly there is cell body, and from the cell body there are some processes come out. Some processes come out of the cell body. So one is long process, that is axon, and others are short processes. These short processes are dendron, and these dendron produce several branches. These are dendrites. One neuron can you imagine how many dendrites one neuron maximally can have? Maximally, one neuron can have maximum dendrites near about 10,000. near about 10,000 dendrites. And the number of neurons in our nervous system, how much? 10 to the power 11. 10 to the power 11. In some reference books, you may get 10 to the power 12. So this is the approximate number of neurons in our body. But what about the numbers of neuroglia? Whether the number will be less than neurons or more than neurons. Their number are much more than neurons, about 10 times more than the neurons. So, the neurons, this is the structural and functional unit of nervous system, send impulses and to and from the CNS, central nervous system and PNS, and to the effectors. This is known to us. Okay. And there are different types of neurons, but most have certain structural and functional characteristics in common. And we know when we study the animal cell, so typically all these things may not be observed in different types of cells in our body. But one thing which are more common to all, that is described in a cell. Similarly, neurons can vary in their structure, in their shape, so they have certain common features. These common features are these. What are these? There will be a cell body, there will be small processes coming out from the cell body. These are dendron, and they will produce number of branches. These are dendrites. And another process that is longer than this, much longer than this, is axon. The point from where the axon originates, it is called axon hillock. Axon hillock. And these axons, they may be myelinated, they may be non-myelinated. They may be myelinated. If the neurons is having myelination, then we will call it myelinated neuron or myelinated nerve. And if it doesn't have this myelination, then we will call it non-myelinated or unmyelinated nerve. Now see, the axon, this axon at its terminal again produces number of branches. Each branch ends in a bulbular structure, bulb-like structure, button-like structure. These are called end button. These actually form synaptic knobs. They will form synapse with other neurons. Or it may, when it knob will be supplying the muscle, they will form neuromuscular junctions. So either they will create synaptic knob. In neuromuscular junctions, they will also form, same knob will also form form synaptic knob. So axons must produce synaptic knobs. Now the neuroplasm, here the cytoplasm is called neuroplasm. And this neuroplasm, when continues through this, actually, what is this axon? It is a hollow cylinder. It is a cylindrical structure. So, at the lumen, what is flowing? Neuroplasm is flowing. So, this neuroplasm present in axon, we call it exoplasm. Now, let us see what is there in the neuroplasm. What you will get? We will get a nucleus. We will get a nucleus. And other than nucleus, what we can get? We can get mitochondria, endoplasmic reticulum. So what is the purpose of the endoplasmic reticulum? Proteins. So they produce proteins. They produce neurosecretions, which we know the neurosecretions Neurons, they produce neurosecretions. These are released from the synaptic knobs. From the synaptic knobs. Now question comes, how these neurosecretions are formed here. How they will go there? Through exoplasm. Okay. This endoplasmic reticulum not only produce neurotransmitters, they also form different types of protein necessary for their own growth, for their survival, for their maintenance. Okay, so these are also formed by the endoplasmic reticulum. So the nerve has to perform a lot of activity. So it must have, it has to generate ATPs. So for that purpose, what is needed? Mitochondria is needed. So these are also having mitochondria. And one of the important characteristics of neuron is that the neuroplasm will contain nasal granules. Now these what are these nasal granules? Nasal granules are actually protein. So these proteins are synthesized in endoplasmic reticulum. Now this endoplasmic reticulum is fragmented and stacked. When this endoplasmic reticulum containing these proteins are stacked along with the fraction of the endoplasmic, they are actually nasal granules. So these nasal granules are present, but you will not find these nasal granules in the exoplasm. You will find it in the neuroplasm, in dendrites. Okay, so beside this, another thing is important for the cell to maintain its shape is cytoskeleton. You have studied cytoskeleton. So neurons have to maintain its shape within the cellular shape. This shape is maintained by the cytoskeletons. So like other cells, they must also have cytoskeleton, like neurofibrils, neurofilaments, microtubules. So these filaments are there. Also, these are also present in exoplasm. Okay? So these are the constituents in nasal bodies. Neurofilaments means cytoskeletons. So there are Golgi apparatus, there are mitochondria. So this area is axon here, and this is the initial segment. Now, if these are myelinated, we have studied, these myelin sheaths are formed by Schwann cells. Schwann cells are actually one kind of glial cell, neuroglia. There are different kinds of neuroglia. One type of neuroglia is Schwann cell. They produce, suppose this is the non-myelinated nerve. This is axon. Inside there is exoplasm, and this membrane is called exolemma. Exolemma. Now what happens? This is a cell called Schwann cell. It is its nucleus. It's a round shape. So one Schwann cell come closer to this. A part of this the axon, the cell, this is the axon, it comes like this. Suppose roti kaisa banta hai? You make a dough and then you make it give it a round shape like this, then then it becomes flat. Now think about the Schwann cell like that. So now this will wrap the axon. Now this will wrap the axons. Both the membranes of the Schwann cell will be coming closer and they will remain attached to it. So both the membranes, so now think the cell, Schwann cell, is flattened, both the membranes opposed to each other, coming in contact, not only they are becoming fixed with the protein. So with this, they are adhered to each other, membrane. Now they will make spiral, spirally they will arrange along the axon. Okay. Suppose these are the two layers, so this will be circulating like the moving like this, and in between these layer fat will be deposited. Now in between these two layers, suppose at the bottom of the another two layer, in between fat will be deposited. So that's why this layer, node of membrane in the myelin sheath, actually it is having lot of fat in it. Proteins are also there, but maximally there is protein. Now where you will get the nucleus? At the outside of the sheath. So they will form so this is the myelin sheath, and length of the myelin sheath is 1 millimeter. And another myelin sheath will be formed here, leaving a gap of 1 micron. Approximately 1 micron, leaving a gap of 1 micron. Another sheath will be formed. So in between the two sheaths there is a gap. This gap is called node of Dendrites. So the whole axon myelin sheath is covered by another membranous structure. This is called neurolemma. Okay? The whole thing is now in a bundle of nerve, there may be lots of numbers of neurons, their axons. So that one axon, information of one axon cannot go to the another axon. For that, so this should be prevented, insulated. So in between there are insulations. In our next class we will discuss it. Nucleus is pale, large, spheroid, and it is located centrally. Neuroplasm is there. It will contain neurofibrils, basal granules, mitochondria, Golgi apparatus, lysosomes, neurosecretory material, cytoskeletal proteins such as neurofilaments, neurotubules, and microfilaments, as I said. Okay, so one thing is very important is there is no like other cells, there is no what? Centrosome. What is the function of centrosome? Anybody? Cell division. So since centrosome is not there, can a neuron divide? No. So neuron cannot divide. So once it is damaged, either it can be degenerated or regenerated. When? It will depend on the injury, whether it will be degenerated or regenerated. Okay, we will discuss. This is degeneration of nerve fiber, injury to nerve fiber. We will discuss. We will learning in our subsequent classes. Basal granules are stacks of rough endoplasmic reticulum. These are proteins synthesized in rough endoplasmic reticulum. Basal granules extend into dendrites but not in axons. They are also present in cell body. So here impulses, they collect, these dendrites collect information from other neurons, and they send it, the cell body receives it, then the cell body send it to the axon terminal. So from this axon terminal, this information will be sent to another neurons or to the muscles or to the glands. So we have already explained it. Now the dendrites, near about 10,000 dendrites can come out of a cell body. The functions of neuron. Now what are the functions of the neuron? So the functions of the neurons, it maintains functional and anatomical integrity of the axon. The neurotrophins, proteins for neurotransmitters, are synthesized in the endoplasmic reticulum of the cell body. We'll learn about neurotrophins, how it is important. Stored in the synaptic knob of exoplasmic flow. Comes from through exoplasmic flow is there. So through this flow, the neurotransmitters formed in the cell body is coming to, actually these neurotransmitters are formed, as I said, in endoplasmic reticulum. So they are packaged. Here they are packaged in a wrapper, that is a membranous structure. So within this membrane there are the secretory granules. Now these secretory granules will roll over to the nerve terminals. Okay, so they are in the packaged. We learn how the information from the axon terminal, from one axon goes to the next neuron using this packaged material, these neurotransmitters. The dendrites, so these are the functions of the cell body, and function of dendrites: receptive process, means it receives information, transmit impulses towards the cell body, and local potentials are developed. Local potentials are developed, means these dendrites, they also connect, each dendrite also connect information from other neurons through synaptic junctions. So these two neurons, these dendrites and axon terminal, there is no direct electrical connection. So this end of the neuron see, there is no continuity. In between there is a gap. This gap is called synaptic knob or synaptic plate. And there is some fluid, extracellular fluid there. So there is no direct continuity. Then how the impulses from here come to here? So it releases neurosecretory substances. And there are, in this membrane, there are receptors. When these receptors binds with these receptors, here some electrical changes take place. Means here a lot of sodium ion will be entering, and this membrane will be depolarized. And when depolarization reach to a limit, threshold level, it will produce action potential, means it will produce nerve impulse. Now this nerve impulse, we said, these are propagatory in nature. Whenever they will find any membrane, they will pass along the membrane. So here, because of this neurotransmitter, one action potential will be generated here. Now this will be transmitted along the dendrites and will arrive into the cell body. And from this cell body, it will be passing to the axon to its own terminal. From there again, it will release neurotransmitters. These neurotransmitters will again will cause generation of the nerve impulses in the next neurons. So in this way, messages are transformed from here to there, from here to there, different organs. Okay. So these are the functions of dendrites. Axon arises from axon hillock, long processes, and we have studied axon. We have already told what are the functions of. Now these axons are of two types. One is myelinated, another is non-myelinated. So we have already told, we will study later on the myelinogenesis, how these myelin sheaths are formed. Okay, so we will study this myelinogenesis, and non-myelinated means there is no, but the central nervous system, there are also some, these are the peripheral nerves are mostly non-myelinated. Long nerves are myelinated, and short nerves in the brain, they are non-myelinated. Moreover, in the periphery, see, this is the vagus nerve, and it will be going to heart or going to intestine or stomach. Okay. So what they will do, here they will have a ganglia. They will have a ganglia. The first they will be terminating here. It will end here. This is the ending of nerve. This is the ending, and this is the cell body origination. Okay. So they will be from here. This is the what? Ganglia. So this is ganglionic cell body. From this ganglionic cell body, another neuron will be formed, and they will form synapses with the myocytes, muscles of the GI tract, and cause stimulation of the myocyte contraction or whatever it is, or it will be ending into parietal cells. They are having acetylcholine receptors. So at their nerve ending, acetylcholine will be released. So now this acetylcholine will be binding with the acetylcholine receptors. When these are stimulated by acetylcholine, thus parietal cells will start to produce hydrochloric acid. So these nerves are very short. Postganglionic nerve. Preganglionic nerves are long, so they will be myelinated. But postganglionic nerves are short, they are non-myelinated. But in case of sympathetic nervous system, the ganglia lies here close to the spinal cord. So preganglionic nerve fibers are short, but their postganglionic fibers are very long. Here postganglionic fibers will be myelinated. Sympathetic, parasympathetic, these are autonomic nervous system, no? For parasympathetic nervous system, they must have some nerves coming out from brain, means cranium, means medulla. We know there are different cranial nerves. One of the cranial nerves is vagus, glossopharyngeal, oculomotor, optic, different types of cranial nerves are coming out. When these cranial nerves are coming out, their cell body remains in the brain, in the thalamus, in the medulla, or in the brain, central nervous system. Only thing, their axons are coming out. Now the axon terminal, where they will be terminating? Where they will be terminating? They will be terminating in a ganglia. And is this ganglia for parasympathetic nervous system? This ganglia lies close to the organ where it supplies. The vagus nerve will be supplied either heart or GI tract or lung. So their ganglia will be present close to the organ. So nerves that are coming out from here, they have to go to the ganglia. So this is a long nerve. So since these are long, these are preganglionic. These are pre. ganglion. Okay, so these preganglionic nerves of parasympathetic nerve are myelinated. But postganglionic nerves, since the ganglia is lying close to the organ, so this will be short. But this is opposite in case of sympathetic nervous system. Why? The ganglia lies here, close to the spinal cord. So from preganglionic fibers originating from spinal cord will end into the spinal ganglia. These are the spinal ganglia. From this spinal ganglia, postganglionic fibers will be supplying the organs. Here, the postganglionic fibers are long, but preganglionic fibers are short. This is one of the difference between sympathetic and parasympathetic nervous system. Another difference is usually from their nerve endings, acetylcholine is liberated. But sympathetic nerves, they are from their nerve endings, it is not acetylcholine, it is adrenaline or noradrenaline. So these are some basic, we will be learning in detail. In non-myelinated nerve, why there is no myelination? Those Schwann cells are there. This is Schwann cell. Normally this way myelination is produced. Now what will happen? This end will continue. So that's why in most cases the Schwann cells remain nearby, but they do not encircle them. This non-myelinated nerve fiber is basically short. They remain mostly in brain, central nervous system, and their speed, the velocity of nerve impulse, the speed with which nerve impulse passes through the axon is different from myelinated. In non-myelinated nerve, the passage of nerve impulse is like this. And in case of non-myelinated, saltatory, jumping, so their speed will be much faster. Because of, why it is faster? Because of presence of the myelin sheaths. We will discuss later. Classification of neurons. How do we classify? Classification of nerves. We will again study the classification of nerves. Neuron and nerves are not same. A lot of neurons, axons, when are bundled together, then we call it nerves. Okay, so these neuron, axons can be classified. How? Depending upon their functions, depending upon the number of poles, depending upon the length of the axons. In different way, through this pathway we can divide them, classify them. So first thing is what? Depending upon their functions. So let's see. We know it. Some neurons will be carrying impulses from the receptor to the brain, from periphery to the brain. So they are what? Afferent nerves or sensory nerves, bringing sensations to the brain. And another neuron that are coming from the brain to the periphery, to the target organs, bringing information from the CNS, what to be done in the affected organ. So these nerves are called efferent or motor nerves. So they are two types. And there is another type, interneurons. So information is coming, entering into the brain or in the spinal cord. So they will be ending in another neuron. Now, one or more neurons. So here it will be decided what to be done. When it is decided, then only it will be sent to the efferent nerves. So in between afferent and efferent, there are what? Interneurons. In between, that are connecting afferent and efferent. Interneurons send information between the sensory neurons and motor neurons. And see, this is the receptor. They are sending impulse to the afferent nerves. Afferent nerves sending information to the interneurons. Interneurons sending information to the efferent nerves, motor nerves. They are ending into the muscles and causes muscle contraction. This way they work, these three. So functionally, we divide the neurons into sensory, motor and interneurons. Gussa kuch? Okay. Afferent, interneuron, this is spinal cord, efferent. Here, affect is put. Either it is muscles or these are gland. And depending on the number of poles, cell body, how many poles it is having? Pole. You don't know pole? How many poles here we are having here? 2. If in a cell body, one pole comes out of the cell body, then it call it unipolar. These are mainly found in invertebrates, lower class animals, in their nervous system. Now, you see, this is bipolar. This is cell body and one pole and a pole. This may produce axon and this may produce dendrite. The cells may have multiple poles. Typical neuron. Multiple poles, most of them are dendrites, and one pole is axon. In some cases, what happens? One pole, from the same body, one pole, and now this pole divides into two parts. Cell body is not producing two poles, one pole. It is divided into two parts. One is peripheral part, one is central part. This peripheral part may produce dendrites, and the central part may produce dendrites, and the peripheral part may produce axons. So these are pseudo bipolar. These are pseudo bipolar. It will be dendrites, so it will appear in pseudo. Actually, these are monopolar. So these are sort of unipolar neurons, cell body, one pole. This is produce central processes, this is processes peripheral. Pole is one. Bipolar, example, dendrite, there's a cell body is producing two poles. In earlier, there was one pole. Now it has divided, this pole has divided. Central part, peripheral part. Multipolar, these are multipolar. Okay. Unipolar, bipolar, multipolar. As I said, this is present only in invertebrates. See, this is pseudo bipolar cells. This is our spinal cord. Here, within the vertebral column, there is spinal cord. So from the spinal cord, nerves are coming out, nerves are entering, nerves are going out. So entering it through our posterior root. We have posterior root. This is the posterior root. These are the anterior roots. Through this anterior root, nerves from the periphery, from the receptor, is coming into the ganglia. We said along the spinal cord there are ganglionic cells. Ganglion. Okay, so there was no scope for discussing